Dear viewers, Assalamu alaikum. Today we will discuss different web servers or online tools which can be used for the prediction of targets for our drugs or compounds. So let's start with the first one, which is Swiss target prediction. It is from Swiss Institute of Bioinformatics and the reference is given here. For this tool, we can use Smiley's file here, or we can sketch a structure, or we can draw a structure here from Marvin sketch, and then we will select our species for which we are going to target. So we can use Homo sapiens, musculus, or Ratus norvegicus. So we can select a species and then we will go to predict targets. After some time we will get our results which gives target for this drug. So here is the carry molecule and here it represents the target classes and the percentage for the class of protein in which it is going to target. Like here the most percentage is 33.3% for enzymes. The color shows that it is the enzyme that is targeted by this compound, and then 13.3% and 13.3%, these are the oxidoreductases or phosphatases enzymes. So we can zoom it and then we can it shows that 33.3% enzymes, 13.3% oxidoreductases, then phosphatases, then nuclear receptor 13.3% also, and then family of G protein coupled receptor, it is also 13.3%, and then electrochemical transporter 6.7%, and the cytochrome P450 is 6.7%. So these are the this pie chart represents the percentage of our target prediction. Now we can save our results, we can export our results in different file formats like PDF, like CSV, or we can also save some other files format. So here it is 15 entries here, and the the first one is the cyclooxygenases. Here is its Uniprot ID, Campbell ID for this protein. This is the enzyme of oxidoreductases target class, and the probability is almost maximum. Non-actives are 3D non-actives are 62 and the 2D are 1. So we can download the all active kind of analogs which are active against this enzyme, and then we can see that our compound is also targeting this enzyme. Then some other enzymes are also shown here, their common name, Uniprot ID, and their probability, and we can download these. What are the 62 compounds? So we can search here. So these are the compounds. So we can take this library of compounds and we can screen for drug-like properties or admitox properties. That last time I have uploaded my video for screening of library, and we can also then perform docking studies for these compounds, and we can select our compound. So so here we have different targets. We can move to other page, and then in this way we can it shows that what are the targets. Now we move to second one. So this is our second target prediction tool, which is Way2Drug. It is a pass online tool. Previously it was pass online. Now it is named with way to drug. So here we can input smileys here, or we can draw a structure here that I have already drawn here a structure, and then we can predict targets for our structure. If we are going to paste here smileys, then we will click on this one, or if we are going to make structure, then we will predict this one. So predict targets for drawn structure. We can also take information about this way to drug, how it works. So it is based on QSAR models, and so it ligand-based multi-target computational system based on a public data and naive-Bayes approach, and also interpretation, how it interprets. So pass target provides the following functionalities. So we can check this, that how it works or how it interprets our results. Now I am moving to target prediction. So predict target, click on it, and we will get our results here. So these are the predicted protein targets for compounds of your interest, and these are the graph. If we move cursor on this graph, it is a family of G protein coupled receptors, and here is the enzyme. Number of target in distinct classes is 21, and then kinases, phosphatases, G protein coupled receptors, and then unclassified proteins are proteases, nuclear receptors, and ligand gated ion channels. So these are the classes which are also predicted by the Swiss target prediction tool. And if we look at here, so these are the signal transduction, anatomical structure development, cell differentiation. So these are the target associated with distinct classes of proteins, and these are the target associated with biological processes like inside the cell, or biochemical pathways or cell structure. Like here, if we look at here, it is the go biological processes. So it is associated with cell differentiation, anatomical structure development, so cell signaling proteins, and then multicellular organismal development. So it is based on that kind of proteins which are present inside the cell, and they have different functions other than the biochemical pathways. List of proteins predicted as possible direct targets. So direct targets are these proteins. So here is the list. We can copy it, or we can save CSV file. So these are the different serine threonine protein kinase, and then dual specific, and then PI4 kinase beta subunits, and so these are the targets. Similarly, we can check that this is the list of the protein predicted as best possible targets. So these are the possible targets, not direct targets. Cyclooxygenases, 0.372% confidence level, and the Campbell ID is this one. So these are the possible targets, and these are the direct interactions or direct targets. So in this way we can find out our target proteins for our compound of interest. Now we move to third target prediction tool, which is LigTMap. So LigTMap also search target proteins for our drugs or compounds, and we will put our smileys file here. So then we can choose this one. I want to type manually, or we can draw a molecule. When we are going to draw a molecule, we will click on it. It will show our sketcher, and then we can draw a molecule here. So this is our point. So we will select benzene ring, then we will move to this point. Our cursor will be moved here, then we will click on it. Now we have made a benzene ring, then we will move to this bond, and now I have pasted here, then another bond, then we can make a double bond here, and then we can select our different elements. Like here I am going to add here bromine. So in this way we will draw our structure. I am going to zoom it. After making structure, we can go to prediction, or we can type manually. So here I am going to paste here my smileys, which I have already pasted in previous two tools for target prediction. So here we have an option of target classes, for which we are going to check that which target class it is going to interact. Like here I am going to check with Helicobacter pylori, so it is the bacterial class, and here tuberculosis, it is also associated with bacteria. So if we are going to hit bacterial protein, we can target these two, we can select these two bacteria, or we can also target here in viruses like HCV virus or influenza virus or HIV virus, or we can select here anticoagulant proteins or beta secretase or carbonic anhydrases or isomerases, kinases, ligases. So all these are the different classes of enzymes. Or we can directly go to bacterial class, that which bacterial class it is going to hit or targeting. So in this way we can select our class of targets for which we are going to analyze that our compound is going to target. Then we will go to here, it is the job description. We can write here what is the job description, and then save our email ID. So here we will put our email ID, and then we will go to check here that I am not a robot, and then we will submit our file. So after the job is done, it will give us results. So it will find targets for 3 different classes, like viruses we have selected, and then we selected a bacteria and TB bacteria, mycobacterium, and then we also selected Helicobacter pylori bacteria. So when the job is finished, we will get our results. We have 3 tasks and it has completed 0 still. So here this compound follows Lipinski rule of 5 and show results here. So we can get results, no results still. So here we will get detail report, and then we can download here. Before this, okay, we will go to another tool, which is molinspiration. It is also a tool for the prediction of our targets for our compound. So I am going to paste here smileys file. We can also draw here a structure for our compound, and then we will go to, we can for molinspiration we can calculate properties of our compound, we can predict bioactivity or galaxy 3D generator. So if we go to properties, we can calculate what are the different physicochemical properties for our compound. So this is our compound. Here is the smileys file for our compound, and here we have log p value 3.75, polar surface area is 63.33, number of atoms 22, molecular weight 293, and the heteroatoms then oxygen and nitrogen 4, and violations of Lipinski rule no, number of rotatable bonds 5, and volume is 264. So we can get data as text format from here. We can get 3D geometry of our compound. From here we will move to predict bioactivity. So directly we will go to select this one, and we will go to bioactivity of our this compound. So more inspiration bioactivity score represents that it is mostly going to target GPCR ligands, G protein coupled receptors, then ion channels 0.05, kinase inhibitor 0.06, then nuclear receptor 0.40. This is also the important one. So this is most important, nuclear receptor ligand. So it has the maximum value is 0.40, protease inhibitor minus enzyme inhibitor is 0.32. So again it is showing that or this compound is going to interact mostly with the nuclear receptors, enzyme inhibitor 0.32, and then G protein coupled receptor, which is 0.27. So this is our tool for the prediction of targets for our compound. Now we are going to check this for our results. So it will take some time because it is also going to check that either. So now we are going to select verify this, then we can move to submit our file. Again I am going to show you that our results. So until that we are moving to another tool for target prediction, which is PPB. PPB is polypharmacology browser for target prediction in Campbell. So this page also works for the target prediction. So here is our compound we can draw here, or we can also PDB ligands or load test ligands. So we will draw here our structure, like here we have drawn this structure, and then we can check that what will be the results for our compound. And number of targets 20, and these are the fingerprints. We can select or unselect all these. So we will go to submit. Okay, now we are going to open LigTMap, and here we got our results for our job ID. So selected target classes are H.Pylori, HIV and tuberculosis. Description, I have changed the description to Majid, then cutoff is 0.4, and here is our input smileys file. So number of targets found for selected target classes. So only from 3 classes only one target class is selected, and this is the HIV. So it is going to target HIV, and target class is only HIV, and it is going to show that it is going to hit one protein, which is the protein data bank, which is 3GIP. It is the PDB ID of this protein which is going to target, and the target class is human HIV virus, which is immunodeficiency virus, and the target name is reverse HIV1 reverse transcriptase P66. The ligand name is P4Y, which is in co-crystallized with this enzyme. So our ligand similarity score is 0.414, binding similarity score is 0.286, LigTMap score is 0.376, and the predicted affinity is 6.163, which is minus log M. And moving towards right, so here we have docking score is minus 5.664 in kilocalories per mole for this compound inside this enzyme, and docking poses we can get here and select for binding similarity analysis. So we can check the docking pose. So here is our input smileys and target class, target protein, target class, and target name, and docking score, and then these are the details, and here is our result, which is going to bind here. So this is our ligand or compound which is going to bind in this protein. So we can it can be shown here. So in this way we can get our results, and it also shows the docking pose of our compound and also the Gibbs free energy values. So we can download here our structure. So we can download the complete docking pose of our compound, and we can analyze in Discovery Studio Visualizer also. So I'm going to open it. So here is our compound, and this is the target class. This is our compound. I am going to change its style to, sorry, I am going to change the display style of this chain, which is our compound, our ligand. So we are going to display style. Okay, now this is our compound which is going to bind here. Okay, so it also gives out us docking results. For molinspiration, I have already shown you that we can produce physicochemical properties of our compound, or we can also check the results for targets. Here it also shows that nuclear receptor ligand is 0.40, which is most probability for this compound. And we have this tool also, PPB, and we are going to submit here number of targets, so it will give us also results. It will take some time. Then we have target hunter tool. So target hunter of small molecule, we can also use this tool for our compound. Like here I am going to paste smileys here for our compound. We can choose file. In a small file we can draw here our structure like previously we have drawn, and then we will go to 2D similarity search. Fingerprints is FP2, bioactivity from literature and bioactivity from PubChem, and we will submit our results. Now this is our compound. For this compound we have these targets and bioactivity, type, value, and the reference from PubChem. So these are our different target proteins with Ki value and IC50 values. So this type of compound going to hit. So this compound is with 0.86 score with Campbell ID, and this compound which is similar to that kind of structure, and it is going to hit with these target proteins, and then it also represents the IC50 values and Ki values for different receptors. So in this way this target hunter of small molecule works. We can return, and now we check the results of PPP, PPB. So here is our results for PPB. Here is our query molecule for PPB, and we are going to check. It represents query molecule, then p value stretching from, then so here we have these targets. So here in green color it represents the p value, and it represents that which target it is going to hit. So these are our different targets, and these are their values. Not hitting values will be in gray, and the green value represents that it is going to hit that targets. So these are different targets it is going to hit. So in this way we get results for our query compound, and we can save our results from here, and this file format will be saved. So in this way we get our results. So all these results are for target prediction. So we can predict target. We have used different tools like Swiss target prediction. We used way to drug. Then we used our this tool, which is LigTMap. It also produced docking results. Then molinspiration, and then PPB. It also gives us the different parameters which are shown here, and then we have target hunter for small molecules. So in this way we can predict target for our drug. So thanks for watching my video. In our next video we will come with some other interesting tools. So thanks.